Good evening, ladies and gentlemen. In this screencast, I'm going to be running you through a process that a company called Quorn, spelled Quorn, produces a product called mycoprotein, and how this can be used as an alternative to meat. And many people these days are choosing to live a vegan lifestyle, whether it be due to the current exploitation of animals or diet requirements. But Quorn have a vision to make the world's diets healthier and more sustainable. So they don't start where everyone else does when it comes to protein production. Rather than using livestock, Quorn produces a natural nutritious fungus from the soil and ferment it to produce a dough called mycoprotein. It's high in protein, high in fiber, low in saturated fat, and contains no cholesterol. And because producing mycoprotein requires less land and water than producing animal proteins, it has a 90% lower carbon footprint and is therefore a lot better for our planet. So I'm going to run you through a brief history of how Quorn came to produce the current product that is now selling commercially worldwide. It began back in the 1960s. Experts were concerned that the predicted growth in the world's population would lead to a global food shortage and widespread famine. And a particular British industrialist named Lord Rank, who was the chairman of Rank Hovis McDougall, the group of companies known as RHM, was founded on the flour milling business and was a major manufacturer of cereals, the main waste product of which was starch. Rank passionately believed that something needed to be done about the impending food crisis and briefed his research director, Dr. Arnold Spicer, to investigate the feasibility of a process to turn starch into protein using some form of fermentation. Fast forward to 1985, and the first ever mycoprotein retail produced was a vegetable pie, and this was followed by the launch of Quorn pieces in 1990, and now there are over 90 products in the Quorn range that are sold worldwide. So what is a mycoprotein? Well, mycoprotein is the ingredient name for a food-grade protein source that has been available for use only since 1985. And as the name suggests, this protein is made up of a type of fungus, Fusarium venetum, and that was found in the wild and developed specifically for the production of food-grade protein. So I'm going to quickly run you through, in detail the production methods of this mycoprotein. So, in its inception, two methods of productions were considered, and they were the fed batch and the continuous flow culture. So the advantage of the fed batch is it's a simple system. It's commonly used in industrial fermentations, and it's used for manufacture of high market value products. Now, the advantages of a continuous flow culture is continuous production of biomass over long periods of time, and consequently, it's comparatively cheap. So the continuous flow culture system was the one that Quorn has gone with. And these next few slides were provided by the company, and it shows you a brief overview of how the actual production works. So, the new fermenters that they are currently using are the largest continuous flow culture systems in the world, each costing about $37.5 million to build. They're 50 meters tall and weigh over 250 tons. Each 155 meter fermenter, 155 meter cubed fermenter, is inoculated with a 5-to-1 of a batch culture containing 50 grams of biomass, and continuous flow is initiated 4 days later. In an airlift fermenter, most of the oxygen transfer takes place near the base of the relatively wide riser, where sterile air is introduced, and where the height of the fermenter creates a high hydrostatic pressure. This, together with the turbulence and consequently small bubble size, provides excellent conditions for the oxygen transfer from the gaseous to the liquid phase. Thus, the riser contains a two-phase mixer of air and culture flowing together so that the air bubbles contribute to 50% of the volume of the fluid. Um, the rate of transfer of oxygen from the gaseous to the liquid phase decreases the culture flows to the top of the riser, where the gas contains about 10% oxygen. The low pressure region at the top of the riser causes a release of CO2, and the culture then enters the downcomer, where at the bottom it is directed into the riser and it is again charged with air, thereby completing the pressure cycle. So the culture is maintained at about 30 degrees Celsius by a heat exchanger set into the downcomer. To prevent oxygen limitation, the downcomer is provided with an oxygen supply. The difference in specific gravity of the relatively aerated culture in the riser and the relatively air-depleted culture in the downcomer, which creates this hydrostatic pressure differential, ensures that the high fill filaments circulate continuously around the fermented loop at a rate of about 1 circulation every 2 minutes. So in the next step, the nitrogen supply for growth, which in this case is ammonia, is fed into the fermenter with the sterile compressed air at the base of the riser. The rate of supply of ammonia to the culture is regulated by a pH monitor, and it's set to give a culture of pH 6 or around that. The nutrient solution is fed to the culture to give a dilution rate in the range 0.17 to 0.2 and is operated as a glucose stat. That is, glucose is always in excess and the fungus always grows at the Vmax at a biomass concentration of about 10 to 15 grams per litre. The fermenter is manned on a 24-hour basis, and tests for the presence of mycotoxins are made every 24 hours. They use a method sensitive to 2 parts per billion, and tests made to date have all been negative, as would be expected for the growth conditions employed. The product is harvested continuously, and Quorn 2 and 3, the code names for some of the fermenters, each produce an outtop of 30 tons. The harvested mycelium cannot be used as it is harvested. So basically, if human food contains too much nucleic acid, the blood uric acid value rises, and the excess accumulates as crystalline deposits in the joints and tissues, leading to the disease known as gout, and it can also lead to kidney stones. So this is a peculiarity of the human metabolism in that the uric acid is produced by the breakdown of nucleic acids, whereas in other vertebrates, sparingly soluble uric acid is converted to highly soluble and allantoin. Because of this, the World Health Organization recommend that the human ingestion of RNA from single-cell protein, such as used in this Quorn's mycoprotein, for adults was defined as about 2 grams RNA per day, and with the total nucleic acid ingestion from all sources not exceeding 4 grams per day. So the mycoprotein biomass cultured at a specific growth rate has an RNA content of about 8 to 9%, which would limit the ingestion of mycoprotein to not more than 20 grams per day. And because of this, a method was developed to reduce the RNA content of mycoprotein whilst minimizing the loss of the protein's fibrous structure, because this was always a concern of Quorn's when changing the conditions of the bioreactors. So in this process, the temperature of the biomass is raised to 68 degrees for about 20 to 30 minutes to stop growth, disrupt ribosomes, and activate the endogenous RNases, which break down cellular RNA to nucleotides and diffuse through the hyphal wall into the culture broth. Importantly, RNases are more heat resistant than proteases, so the protein loss is minimized. This method is carried out in the culture broth with no other adjustments, since a pH of 5 to 6 is optimal for RNase activity. RNA reduction has a substantial economic penalty attached to it, since as well as removing RNA, other cell constituents are inevitably lost during this process, including up to 30% of biomass dry weight and a significant amount of protein. but it is essential to reduce the RNA content. So this step had to be done. After this treatment, the microprotein was shown to contain only 1% of RNA, and that's similar to the present in animal liver and well within 2% range. So the biomass made up of colonial mutants possesses the same chemical composition and is nutritious as the parental strain, but presence of the colonials changed the texture of microprotein quite a bit, and it causes it to become a bit more crumbly. So because of this, Quorn decided to do a few tests on the mutant types to try and come up with a product that had the most feasible and the most desired texture of this protein. So the culture needs to be ended, and a new one needs to start when the clones start appearing, because the appearance of colonials caused the premature termination of the fermentations, and consequently the loss of productivity. When grown in plate culture, highly branched mutants from the colonies, which are much more compact than those of the parental strain and expand in radius much more so than the parental strain. This is why they're called colonial mutants. Despite their reduced radial growth rate in turbidostat or chemostat culture, at a high dilution rate, colonial mutants rapidly supplant the parental strain. So after emerging from the fermenter, the microprotein is subjected to a temperature of 65 degrees Celsius, a treatment which triggers the breakdown of most of the fungal nucleic acid, and the level of which otherwise exceed health and safety limits. So the material is then dried, frozen, and when it emerges from the dryer, it's said to have a similar look to pastry and has a slight mushroom-like smell to it. It's then processed into food and food products, which usually contain added flavorings and other ingredients to make the product taste more desirable. Microprotein production represents an efficient use of cheap and readily available energy source, and the harvested product readily absorbs a wide variety of flavorings and coloring. Whilst its fibrous texture arising from the mycelial origin of the fungus makes it easy to formulate into products which they're said to chew like meat and fish. And many people these days say that it's a great alternative to all meats. We need to know the risks of production. So with all revolutionary foods, microprotein has come under scientific scrutiny. And two studies were done, one in 1993 and one in 2002, which produced results that suggested that the microprotein had potential negative effects on certain subjects with allergies to mold. The 1993 study found that the microprotein had similar allergenic determinants and the Aspergillus fumigatus and Cladosporium herbrum, and some with the Alternaria alternata. They're all basically different forms of fungi. So to test the hypothesis, microprotein production workers were monitored over a 2-year period, and only 2 of 10 patients who were admitted to the hospital because of microprotein ingestion had skin prick tests, but none had significantly raised. These results suggest that the microprotein is not in fact a serious allergen as other fungal pathogens, and therefore bodes well for the widespread microprotein consumption. Thanks very much for this presentation.